Hi everyone, I'm Ibrahim El-Sadek, Computational Optics Group, University of Tsukuba. Today I'm going to talk about optical coherence tomography-based tissue dynamics imaging method for cross-sectional and three-dimensional evaluation of tumor spheroid activity. Through this project, what we are interested in is the optimal selection of anti-cancer drug for individual patient. Recently, a patient-originated tumor spheroid-based drug selection have been released, where we can extract a tumor cell from a real patient and cultivate it as a tumor cell spheroid, and by applying different kind of a drug to this spheroid and check the spheroid morphology, we are going to select the anti-cancer drug and inject it to the patient. And this method is effective but is time consuming. So if we have a more sensitive imaging modality, we are going to optimally select the anti-cancer drug in a few hours or days after drug application. From this perspective, we can clarify which properties we need in our imaging modality. At first, spheroid is a 3D structure, so our method should provide three-dimensional measurement. In addition, spheroid have a few millimeter diameter, so our method should have a sufficient penetration depth. Also, longitudinal study is required, so our method should be non-invasive. There are several conventional methods have been used for tissue viability imaging, such as fluorescence microscopy, which uses an exogenous contrast agent and provide 2D images and have around 100 micrometer imaging penetration. Direct visualization of a subcellular motion have been also released, which is a non-invasive, two-dimensional and have a few micrometer imaging penetration. And recently, optical coherence tomography microscopy have been released, which is a non-invasive, three-dimensional and have a few millimeter imaging penetration. However, it doesn't have a sensitivity for tissue dynamics imaging. So, our purpose in this study is to present an OCT-based method for quantitative evaluation of tumor spheroid activity. Let me talk about our methods and system. Based on the temporal fluctuations of the OCT intensity of a rapidly acquired OCT time sequence, we proposed our log intensity variance method LIV, which is the time variance of the log scale intensity. And why we take a log? To explain about this, let me model the intensity as I equal SD, where S is the static signal magnification and D is the signal dynamics. And once we take a log and compute the variance, we are going to get LIV, which is sensitive to the magnitude of the fluctuation of the dynamic signal component. Our second method is the OCT correlation decay speed, OCDS. And why we introduced OCDS? It's simply because of the limitation of LIV. To explain about this, let me introduce these two signals, which have the same fluctuation magnitude but different fluctuation speeds. So these two signals are going to give us the same LIV. However, the dynamics behind is totally different. So we proposed our OCDS. By shifting the fast fluctuating signal in time and compute the autocorrelation, we are going to get a fast decorrelation curve like this. And by fitting the slope of this curve in early delay times, we are going to get early OCDS, OCDSE. And for the slow fluctuating signal, we are going to get a slow decorrelation curve like this. And by fitting the slope of this curve in a late delay times, we are going to get late OCDS, OCDSL. So OCDSE is sensitive to the fast and slow dynamics, while OCDSL is sensitive only for the slow dynamics of the tissue. In this study, we have utilized our previously built 1.3 micrometer swept source OCT system with a 50 kHz acquisition speed and 19 micrometer and 14 micrometer lateral and depth resolution. And for the acquisition and analysis, we acquired 350 frames in a 4.4 seconds for cross-sectional imaging, while for a three-dimensional imaging, we repeated raster scanning 32 times at each location in a tissue in a 6.5 seconds. To validate our method, we organized three studies. First study is a longitudinal evaluation of tumor spheroid activity, and second study is a visualization of a spheroid's anti-cancer drug response, and then we did a three-dimensional dynamics imaging of tumor spheroids. So, let me talk about our first study. Here we have utilized a human breast adenocarcinoma spheroid, MCF7 cell line, where we seeded the spheroid at day 0, and after 15 days we extracted the spheroid from the cultivation and start the OCT measurement every 2 hours up to 28 hours. Here I have to mention that during the cultivation, the spheroid have been cultured using a CO2 supplied environment, while OCT measurements have been done in a room temperature without CO2 supply. By observing the time course OCT intensity, we cannot see a clear difference in the tissue structure, but once we computed our LIV, we can see that the outer shell of a spheroid shows high LIV, while the inner part shows low LIV. It was reported that the cultivation of a large spheroid exhibits a necrotic core, while the outer shell shows a proliferating cell layers. So maybe the high LIV appearance at the outer shell of a spheroid is related to the proliferating cell layer due to the sufficient nutrient supply, while the low LIV at the spheroid core is related to the tissue necrosis. And by observing the time course of the LIV, we can see that after 28 hours, almost all the spheroid region shows low LIV. Our second contrast is the OCDS-L, which shows similar appearance to the LIV at 0 hour and 28 hours. And by comparing OCDS-L and LIV, we can see that the boundaries between high and low OCDS-L regions is clearly appeared in an OCDS-L image. That's maybe because of these two methods are sensitive to different aspects of the tissue dynamics, such as OCDS-L is sensitive to the slow dynamics of the tissue, while LIV is sensitive to the magnitude of the fluctuations of the OCT signal. Our third contrast is the OCDSE, which shows high OCDSE appearance at the spheroid core. It was reported that tumor spheroid core exhibited fast decorrelation of the OCT signal, which is consistent with our OCDSE image. And by observing the OCDSE after 28 hours, we can see that almost all the spheroid region shows high OCDSE appearance. So let me summarize our three contrasts at the spheroid core. The combination of low LIV, low OCDS-L, and high OCDSE can be called as a tissue necrosis signature. So let me talk about our second study. Here we have utilized MCF7 spheroids and the medication was Taxol. We seeded the cells at day 0 and after 4 days we applied the Taxol, and in day 5 we applied a fluorescence antibody and then measured the sample using fluorescence microscope and OCT. Here we have prepared 3 samples, control, 5 nanomolar and 500 nanomolar treated spheroids. This figure summarizes the results of the fluorescence microscope and dynamic OCT. In a control samples, we can see a clear necrotic signature at the spheroid cores, which exhibits a low LIV, low OCDS-L, and high OCDSE. In 5 nanomolar case, we can see a clustering of the necrotic signature at the spheroid core, which may be related to the dead cell clustering in a fluorescence microscopic image. In addition, a necrotic signature is appeared at the outer shell of spheroids, which may be related to the drug application. In 500 nanomolar case, we can see that the necrotic signature is clearly pronounced at the outer shell of spheroids, which is consistent with the fluorescence microscopic image. It was also reported that the high concentration of tamoxifen or Taxol can induce the cell necrosis of the MCF7 cell lines, which is consistent with our findings. So let me show what we achieved in our imaging modality. We have measured the spheroids longitudinally up to 28 hours non-invasively. In addition, we measured the spheroid samples with a high imaging penetration depth. So what we still need is a 3-dimensional imaging. Our current scanning protocol acquires 350 frames at the same location in a tissue, which is not realistic for 3-dimensional imaging. So how many number of frames we need for tissue dynamics imaging? In our recent publication, we made an optimization analysis where we created the dynamics image using a different number of frames and total acquisition time window. And this figure shows that 17 or 33 frames are sufficient for tissue dynamics imaging as far as the total acquisition time window is large. Based on the optimization analysis, we made a custom-made 3-dimensional scanning protocol where we divided the emphasis plane to 8 groups, and each group contains 16 locations, and these groups have been scanned using raster scanning, and this scanning is repeated 32 times in a 6.5 seconds. So finally, we obtained a volume of 128 locations in a 52 seconds. In this study, we have utilized the MCF7 spheroids, where we seeded the spheroid at day 0, and after 4 days we extracted from the cultivation and measure it using the 3-dimensional scanning protocol at 0 hour and after 20 hours. At 0 hour time point, this figure summarizes the volume rendering of the LIV and OCDSL. A clear necrotic signature is appeared at the spheroid core, while the outer shell of a spheroid shows high LIV and OCDSL. The important point here is that cross-sectional views can be obtained at any direction, and the emphasis planes can be compared to the standard methods such as fluorescence microscopy or histology. After 20 hours, we can see that almost all the tissue regions shows necrotic signature. In addition, based on a thresholding of the LIV and OCDSL, we computed the necrotic cell ratio, which is the necrotic cell volume divided by the total cell volume. So we can see that at 0 hour, the necrotic cell volume is around 30%, while after 20 hours, it becomes around 65%. So let me conclude my talk. The proposed dynamic OCT methods are valid for longitudinally visualizing and quantifying the spheroid activity, and dynamics imaging method visualizes the drug response of the cell spheroids. And our method can visualize and quantify the 3D tumor spheroid activity, and emphasis images can be compared with standard methods. And the presented method is totally non-invasive, so it might become a useful tool for precision medicine research. Here I would like to thank my colleagues, collaborators, and our funding agencies, and thank you.